topic for today is Alzheimer's disease. Let's get the notes. So I'm going to use Google Documents for this quite a lot, just so we can keep track of where we are. It's quite a complicated paper that I want to take you through, and I want to do quite a bit of background before we get into the paper anyway about the disease, just so that everyone's on the same page. So it is 9 o'clock, let's begin. So let's give an overview of Alzheimer's disease. You've probably heard of Alzheimer's disease before. It is one of the more common neurodegenerative diseases. Effectively, you might consider this to be a pre-senile dementia. Just pulling up the Wikipedia article here just for ease of use. If you want to just get the overview, this is absolutely fine to use. So signs and symptoms, it's effectively progressive dementia. Early signs and symptoms, learning and memory loss, small percentage difficulties with languages, executive functions, and it progresses into moderate degeneration that can hinder independence, speech difficulties, behavioral difficulties, changes in personality, and then you have advanced and final stages of Alzheimer's disease, which is effectively the patient is completely dependent on other people, generally complete loss of speech and bodily function. So it's quite a devastating disease. It's quite a scary disease, really, because at the moment there's currently no treatments available, and there's not very good early detection of this disease. So typically with a lot of dementias and a lot of neurodegenerative diseases, they can only truly be diagnosed on post-mortem. So that's where you'll have your brain examined after you die, and then they'll find evidence of certain pathologies within the brain that would lead them to believe that it's a certain type of disease. Alzheimer's is definitely an example of this. Another example of this is Parkinson's disease. Although the symptoms of Parkinson's disease are quite readily observable when the patient is in early onset of disease. So what I'm going to do is, so very simply, so you're going to have to hear me typing neurodegenerative. So we've got a neurodegenerative disease. What I'll do is let's bold these up. Cognitive decline. Progressive. And generally it's earlier onset than kind of your senile dementia. So I think generally Alzheimer's disease most commonly kicks in about the age of 60 to 70, I do believe. So get back down to here. Early onset dementia. So one of the key things is post-mortem diagnosis. And under current treatments, sadly, none at the moment, currently. So if we go into the pathology of Alzheimer's disease, so we talked about that post-mortem evaluation. What you generally find in Alzheimer's disease is you find the presence of what are called plaques in the brain tissue. So a plaque is effectively just an aggregated protein lump that shouldn't really be there. So in all the cells in your body, proteins are produced and then they're degraded. It's kind of like a cycle, and it's all kept in equilibrium. Words are difficult. And when this equilibrium breaks down for some reason, what you find is you get aggregation of certain proteins, but not others. And these aggregations can inhibit the normal functions of whatever that tissue is supposed to do. So in the brain tissue, obviously, aggregates can cause disruption of neuronal signaling, which can in turn lead to things like cognitive decline. And I've got an image of it here. I'll try and zoom it in. You can see it. Where did it go? Here. So what we've got, this is a section of brain tissue. On the top right left hand side, you've got, this is what a normal piece of brain tissue looks like once it's been sectioned and viewed under a microscope. And then you can see with Alzheimer's disease, you get these, they're stained red, but generally they'll be colorless. But there are these plaques that form of different proteins. So plaque formation. And these plaques, generally, they're formed out of two proteins. So there are a couple of hypotheses as to which ones are more important, but two general types of plaque are seen in Alzheimer's disease patients. These are plaques of a protein called tau, and another one called beta amyloid. And the paper that we're actually going to be looking at is one that is going to focus on beta amyloid. I think. I may have a link for beta amyloid here. Cleavage. No, I don't. I won't try and find a picture because it'll probably take me ages. So, but effectively, these are. We'll go into what beta amyloid does in a minute. So these are the two major plaque-forming protein aggregates you see in Alzheimer's disease. Okay, so there aren't any current treatments, but we've known about Alzheimer's disease for quite some time. So what is hindering the development of any kind of therapeutic for Alzheimer's disease? And it really boils down to the fact that it's really, really difficult to study Alzheimer's disease. And for one of the reasons that I've already mentioned, which is the fact that you generally only get a diagnosis post-mortem. And the reason that you only get a diagnosis post-mortem is because you can't sample the brain tissue very easily. If the only way to diagnose Alzheimer's disease is to have a look at the brain, you have to take a bit of the brain out in order to properly diagnose it. We don't have many options in terms of in situ visualization of these plaque formations at the moment. So a lot of the scans that we do have are very expensive, so they can only be done in certain instances. The other issue is that actually this is kind of an issue with almost all neurological diseases or neurodegenerative diseases, is there's a distinct lack of what a scientist or a biologist would call a piece of control tissue. So if you think that you want to have a look at a patient with Alzheimer's disease or any neurodegenerative disease versus a healthy individual, and you want to compare the brains of those two, not only do you have to have a piece of tissue from an Alzheimer's disease patient, which is hard enough to get, but you also have to have a piece of tissue from a healthy disease patient. A healthy patient, not deceased. A healthy patient. And people don't readily give up bits of their brain for research. So the number one difficulty is actually a lack of tissue. Right? I would almost stake my house on it, being that if there was an easy way to get neuronal tissue, then there would be far more advances in neurodegenerative disease biology, for sure. So we can't get actual tissue to study. So what can we do in lieu of that, right? So biologists and biochemists and people that work in this field, what they tend to do is they tend to use models of disease. Now, the term model might not come up in the context that you know it very well. Effectively, what we want to do is we want to build a system of usually cells in a controlled environment that we can test things on. And there are some very good models out there. You know, there's a lot of cell culture models which are utilized all over the world, and we've been using cellular growth models since the 50s. Models around neuronal tissue are a little bit harder to come by because generally models involve the use of what we would call primary tissue or primary cells. These are cells that are taken directly from that organ from a live patient or a newly departed patient. Whereas, again, cerebral tissue is actually very difficult to come by. So the models are quite restricted. So what we can do is we can use what are called cancer cell lines. So cancer cells, they are ever proliferative. So they'll just keep growing and growing and growing. As you know, that's how tumors form. And what we can do is we can take, say, brain neuronal tumor cells. If, say, a cancer is resected out of a patient, and you can grow those cells in a dish, and they'll just carry on growing under the right conditions. So that's an option, and we can study those. But of course, you know, they're a cancer cell line. They've changed immeasurably because they're cancerous. They're effectively not acting normal in the first place. So they're hardly a good surrogate for normal, healthy tissue, or indeed, you know, a neuron that's become cancerous will not behave like an Alzheimer's neuron, if that makes sense. So we have them, but they're not that good. We have something which I am going to talk about in a minute, which is stem cells. You have to apologize for my horrific spelling mistakes that are probably going to be all over this. So stem cells are really interesting. I will go on to stem cells in a minute because that's actually what the paper uses that we're going to have a look at. So that will become a bit clear. Stem cells can be... Yeah, I mean, stem cells, if you don't already know, stem cells are cells that can become other cell types. So if you think that, you know, when you're like a fertilized egg in the womb, you're a single cell. There's one cell there. And that cell divides and divides and divides and divides. And eventually it becomes every cell in your body. Now, you know that your skin cells are different from your liver cells, for example, but back a few thousands rounds of division prior to that, it was only a single cell, and that's a stem cell. And we call those stem cells pluripotent, which is a horrible word to say and to spell. And effectively what it is it just means that they can become any cells in the body. So we can utilize those for models. And the great thing about stem cells is that for a few years now, we've actually been able to generate stem cells. So you might ask, well, where do you get the stem cells from? Well, a few stem cells will exist in your body to facilitate tissue regeneration, just as your tissues kind of get injured or they break down over time. But what we can do is we can make them. So hopefully be able to see this. So this is actually now a very standardized thing. So what we can do is we can take a variety of different cell types. In this example, they've taken fibroblast cells, which is like a connective tissue type cell. And you put these cells, you extract these cells out of a person, and you do what's called reprogramming them. And so reprogramming them effectively puts them back into this stem cell like form. And I say back because it's like you've got the end cell and you want to regress that cell back to when it was like an embryo, like a fertilized embryo. So you can reprogram these cells. It's actually a fairly standard way of doing it now. You only have to add a few factors into the growth media, and these cells effectively regress all the way back to what are called IPS cells, or iPSCs, which stands for induced pluripotent stem cells. Now these things are quite powerful, right? Because now you've got a stem cell, and dependent on what kind of conditions you treat these stem cells, you can turn these cells into other cell types. You can redifferentiate these cell types. And so there's an example of it here on the screen. They've taken fibroblast cells, they've reprogrammed them back to iPSCs, which are in the middle. And you can take these cells and split them up into different dishes and treat them with different media, different growth factors, and you can induce them to become cardiomyocytes, adipocytes of fat cells, dopaminergic neurons, neuronal cells, motor neurons, pancreatic cells, and hematopoietic progenitor cells, so blood cells. So that's quite a powerful tool, and that's what these guys have done in this paper. So these are iPSCs, and these effectively cells that can become almost any cell. So you can see that if you've got these cells, you could reprogram them into a neuron, and then you can have a look at some of the neuronal functions. So you effectively could have a brain-like structure in a dish using these cells. So it's kind of cool. The last thing I want to talk about before we get into the study is another thing called an organoid. Oh. It's an organoid, right? So these are fairly new as well, relatively speaking. So when we're culturing cells in the lab, generally you can grow them as what's called a 2D layer. So it's like a lawn of cells. You have a dish. It's a flat dish, and you put the cells on top, and they stick down, and then they grow in a 2D layer. So they're not stacked on top of each other. They're just next to each other, and they grow and fill the dish, and then you can treat the cells with whatever you want to treat with. Okay, that's a 2D culture system. If you go one step beyond that, you can have what's called a 3D culture system, where you induce the cells to kind of sit on top of each other, right? And so they're still in a layer, or sometimes they're in a layer. Sometimes they're in a ball. If you culture them in a flask that has like a curved base to it, all the cells settle down into the bottom, and they form into this sphere, right? A sphere-like, anyway. And if like the chunk of cells that you're growing has multiple different cell types in there, you generate like a very, very tiny mini organ of a body. And so you can do that. You can make liver organoids. So you can make little liver spheroids that have loads of different cell types that are generally found in the liver, right? And you can do that for a lot of different organ types. So in the paper we'll go into, what they've done is they've made what are called cerebral organoids, right? So these are, just to write this down, these are multicellular culture system. The multicellular culture systems with multiple different cell types in it. No. Speaking's hard, typing is harder. Thank God for spell checker. So what they've made is they've made this thing called a brain organoid or a cerebral organoid. Here's an image of one. Can we blow this image up? Right, this is a fluorescent microscope image. It doesn't really tell you a lot, the structure, but effectively this is an organoid of neuronal cells and another cell type that's found in the brain called an astrocyte. And what you can do with these is you can show, I think they haven't shown in this paper, but in previous papers they've shown that these organoids have the capability to send and receive electrical impulses as a neuron actually would. So you got to remember that these are not perfect. These are not brains. These are not living, but they do have some of the characteristics of neuronal tissue, which is cool, which is a lot better than what we can do without them, which is basically nothing. So, or you can get into animal models of neuronal diseases, things like mice models and other rodent models. But we're not going to go down that route today. Hey guys, thank you very, very much for watching. This was originally broadcast on Twitch. If you'd like to come over and check out my stream Monday, Wednesday and Thursday evenings around 8 or 9 UK time. And I stream a variety of different things, so it'll either be scientific content like this, I'll either be playing some games, or I might be playing a little bit of music. You can see the guitars in the background. So come over and check it out. Thanks for watching.